Welcome to the deep dive. Imagine inventorying, well, pretty much every mammal, bird, and amphibian in a forest, just by collecting the air. Today, we are deep diving into a really revolutionary study that could sort of turn conservation monitoring on its head. Airborne environmental DNA.
It's genuinely groundbreaking stuff. For decades, conservation work has relied on methods that are often grueling, expensive, and frankly, not always that effective. Things like setting camera traps, doing visual surveys, or collecting scat samples.
Right, and when you're facing a global biodiversity crisis, those methods just can't keep up, can they? They're often localized, they're slow, and they notoriously miss the species that are rare or shy or maybe only come out at night.
Exactly. And the solution, well, according to the paper we're dissecting today, it might literally be floating all around us.
Yes, we're diving into airborne environmental DNA. Captures terrestrial vertebrate diversity in nature, a really crucial piece of research led by Christina Lynggaard and her colleagues. We already know that environmental DNA, eDNA, the microscopic stuff shed by organisms like skin flakes, saliva, waste, it works brilliantly in water and soil. But this study really confirms the potential of airborne eDNA, these bioaerosols, for mapping biodiversity in a complex natural environment.
And that jump from a controlled setting to the wild, it's not trivial at all. Previous work, the successful stuff with airborne eDNA, was mostly done in confined spaces. Think urban zoos.
Okay, yeah. Zoos make sense.
Right, because the density of animals is just astronomically high there.
Huh?
Plus, all the human activity probably helps stir up the DNA dust,
Okay.
So taking this technique out into a mixed forest where animals are much scarcer, they're moving freely, and vegetation can actually hinder dispersal, maybe even scrub DNA out of the air. That's the huge scientific leap this paper represents.
I want to pause on that for a second. Why does this matter so much right now? We know vertebrates are essential, but maybe remind us quickly. What's their practical role? How do they keep ecosystems ticking over?
Well, vertebrates are, you could say, the planet's maintenance crew. We're talking mammals, birds, amphibians. They really drive ecosystem health. They're responsible for, well, everything from pollination and making sure seeds get dispersed properly, getting new trees and plants established.
Right, the groundwork.
Exactly, all the way up to top-down predation, controlling populations, even limiting disease vectors. When biodiversity drops, these critical services start to fail. So the need is urgent. We absolutely need unbiased, cost-effective monitoring tools, like yesterday.
Okay, so they proved it could work in a zoo environment. What were the specific hurdles, the logistical challenges they anticipated when taking it out into nature? Specifically, this mixed forest in Denmark they used.
The biggest hurdles are really dispersal and just scarcity. In a zoo, the source, the animal DNA, it's abundant, concentrated. Out in nature, the signal is much weaker, much more diffuse. So the researchers' mission was really threefold.
Okay.
First, just prove the technique actually works despite that low animal density. Second, figure out the best ways to collect it in such a variable and natural setting. And third, start to understand how this airborne eDNA actually moves. Is it staying put or is it traveling around?
Let's get into the nuts and bolts then. The field work happened in Olsen Nature Park in Denmark. You said the sampling period was short. How short?
Astonishingly short, actually. They only sampled over 3 consecutive days. That's it, September 28th to October 1st, 2021. They set up shop across 6 different sites, making sure to cover different microhabitats, like a forest edge near a stream, a more semi-open area, and then a spot deep inside the closed canopy forest.
Trying to see if habitat made a difference.
Precisely. Would slight variations in vegetation or maybe being near water affect what DNA they captured?
And what were they using to actually, well, suck this invisible DNA out of the air?
They used these custom-built air samplers. Nothing too fancy, really. Think simple devices fitted with specialized glass fiber filters, F7 filters, actually. Kind of like what you might find in a good HVAC system. And they strapped these samplers to trees, maybe 1.5 meters off the ground, trying to get them right in that zone where you'd expect bioaerosols to be most concentrated.
Okay, and here's a really interesting part of the method. They tested 2 different airflow rates side by side. Why? What were they hoping to learn there?
Yeah, this comparison was absolutely key. They wanted to understand how sampling volume affected detection. So they tested a low airflow rate, pulling in about 1.1 cubic meters of air per hour. Over a 12-hour sampling event, that's roughly 13 cubic meters.
Yeah, okay.
And then in parallel, they ran a high airflow rate, 3.5 cubic meters per hour. That adds up to 42 cubic meters over the same 12 hours.
Wow, that's more than 3 times the volume of air.
Exactly. More than 3 times. And they did this across 6 12-hour periods alternating between day and night sampling.
Now, eDNA, it's notorious for contamination risk, right? Trace amounts. The lab work must have been incredibly rigorous. What kind of precautions did they take?
Absolutely extreme. We're talking precautions similar to what you'd see in labs studying ancient DNA, which is notoriously difficult to work with. They used strictly unidirectional workflows. Things only move one way through the lab process. Specialized clean labs, dedicated footwear, airlocks, the whole 9 yards.
Wow.
Plus really strict decontamination with bleach and UV light. And get this, to really guard against any contamination picked up after sampling, they even cut off the outer edges of the filters before extracting the DNA.
Removing anything that might have settled on it during transport or maybe got wet in the rain.
Precisely. Anything extraneous.
And they didn't just rely on one way to find the DNA, did they? They used two different search engines, so to speak. Can you explain these complementary primer sets?
Yeah, that's fundamental. You're not just looking for DNA in general. You need specific tools, primers, to target regions of DNA that are conserved enough to amplify, but variable enough to tell species apart. They use two main sets targeting mitochondrial DNA. One, targeting the 16S gene, is generally really good for mammals.
Okay.
The second targeted the 12S gene. That's a broader vertebrate marker, and in this particular study, it turned out to be crucial for picking up most of the birds and amphibians.
And humans shed DNA absolutely everywhere, so they also had to use something called human blockers, right, during the amplification step.
Exactly. Even with all the clean lab protocols, human DNA is just unavoidable. These blockers basically work by preferentially binding human DNA during the PCR step, that's the polymerase chain reaction, how you make millions of copies of the target DNA. So the blockers essentially prevent the human DNA from overwhelming the signal from the wildlife DNA they actually want to sequence.
Okay, so after all that incredibly careful work, collecting the air, the rigorous lab protocols, the different primers, what did they find? What were the big results?
Well, the results were genuinely staggering, especially considering the time frame. In just those 3 days of sampling, from only 143 filters, they identified 64 non-human vertebrate taxa.
64.
In 3 days, 64. And 57 of those were considered truly wild species native to the area. To put that into context, they detected DNA from over a quarter more than 25% of all the estimated 210 terrestrial vertebrates known to live in that entire region of Denmark.
Wow. Over a quarter of the local fauna, just from sampling the air for 72 hours. What kinds of species did they detect? Was it mostly birds or?
It was incredibly diverse. Birds were the most common group detected, species like the common pheasant, for instance. But they also found mammals, amphibians, and rather surprisingly, even two species of ray-fin fish, the stone loach and the gudgeon.
Fish. How does fish DNA get into the forest air?
Right. It's likely not aerosolized directly from the fish themselves. The thinking is it was probably transported maybe via predators like birds or mammals that had eaten them near the stream, or perhaps even aerosolized off the stream banks during windy conditions.
That's fascinating, and it highlights something else important. This method seemed particularly good at finding species that traditional methods often miss.
That's really the power of eDNA, isn't it? It's a more unbiased sample of what's actually there. They picked up signals from the nocturnal Eurasian badger, for example, and the woodcock, which is notoriously elusive.
Things you wouldn't easily spot with a camera trap or a visual survey.
Exactly. And even more intriguing, they detected 4 taxa that weren't actually known to live right there in the immediate study area, but are found elsewhere in Denmark. This included the scoter genus, a type of sea duck that migrates over land, often at night. So it suggests airborne eDNA could even track animals moving through an area, not just living there permanently.
Okay, this brings us to what I think is one of the most novel findings, methodologically speaking, the airflow mystery. You mentioned the high-flow sampler collected over 3 times the air volume, but it didn't detect significantly more species than the low-flow one. That seems counterintuitive. Why is that unexpected, and what does it actually tell us?
Well, you'd logically assume that if the DNA just sort of evenly mixed in the air, like a fine mist, then sampling more air should absolutely mean capturing more DNA, and thus more species. Simple probability.
Right. More air, more chances.
Exactly. The fact that it didn't work out that way strongly implies that airborne eDNA isn't evenly distributed at all. It's not like a smooth, uniform cloud. It seems to exist in these scarce, maybe short-lived, localized patches, probably linked to specific shedding events or very localized air currents.
Huh. So if you're designing a monitoring program, the takeaway isn't necessarily build a bigger vacuum cleaner.
Not at all. It suggests successful sampling might depend much more on clever strategic placement of your sampler than on simply maximizing the volume of air you process. It shifts the focus, perhaps, towards understanding micrometeorology and animal behavior to predict where those transient patches of DNA might be found, rather than just brute force sampling.
So placement over power,
Yep.
essentially.
Precisely. It suggests maybe using more, smaller, less power-hungry samplers placed strategically might be better than fewer, larger ones. And interestingly, when they looked at the spatial distribution between their sampling sites, forest edge versus closed forest, they found the detections were pretty much randomly distributed.
Meaning the DNA seems to move around quite a bit within the forest.
Yeah, it implies that once the eDNA gets airborne, it can travel relatively easily throughout the local environment, sort of blurring those specific microhabitat signals over the timescale of their sampling.
Okay, so spatially, things seemed mixed up. But temporally over the 3 days, they did see a pattern, right? A systematic shift in the detected community.
They did. There was a clear temporal trend in the composition of species they detected across the 3 days. But interestingly, it wasn't a simple day versus night difference.
So not just nocturnal animals showing up at night and diurnal ones during the day.
Not clearly, no. Instead, when they looked at the weather data, the shift in detected species correlated strongly with meteorological changes. Specifically, an increase in rain and wind speed, along with a drop in atmospheric pressure, seemed to coincide with changes in what they found.
Ah, so the weather itself is influencing the sampling. Rain washing the air clean.
Essentially, yes. It's a phenomenon called wet scavenging. Wind might stir particles up initially, but then increased rain acts like a giant atmospheric scrubber. It washes particulate matter, including the eDNA attached to it, out of the air. So a heavy rain event could significantly alter what DNA you detect in the air immediately afterwards.
Okay, so pulling this all together, what does it mean for the immediate use of this technology? It sounds incredibly sensitive, efficient, needs relatively little sampling effort.
I think it firmly positions airborne eDNA as a really valuable tool. It can help generate biodiversity information much faster and potentially more comprehensively than traditional methods, especially for those hard-to-find species. It's a powerful complement to existing approaches. But the study was also crucial in highlighting the inherent challenges you face when working with such tiny amounts of DNA from the environment.
Right, let's talk limitations. Dealing with DNA in such low quantities, what are the main headaches?
Well, the first big one is what's called low-template DNA. Because you start with so little DNA, you have to run the PCR amplification for many, many cycles to get enough material to analyze. And doing that introduces something called stochastic amplification.
Stochastic meaning.
Random. Exactly. Think of it like trying to photocopy a page with only a few words scattered on it. In one copy attempt, you might get certain words. In the next, you might get slightly different ones, or miss some entirely. With low-template DNA, the amplification process can be random. A species' DNA might get amplified successfully in one technical replicate, but be missed in the next. This can lead to false negatives, or potentially even false positives, if trace contaminants get amplified.
Okay, that sounds tricky to manage. And what about contamination? You mentioned human blockers, but it still seems like a major issue.
It was, yeah. Even with blockers, human DNA sequences popped up in, I think, up to 44% of reads in some samples. That's substantial. But the really dramatic contamination came from pigs.
Pigs in a nature park.
Yeah, the 16S mammal primer set in particular frequently got overwhelmed by pig DNA. In one sample, pig sequences made up literally 100% of the reads.
100%. So you find nothing else.
Pretty much. It completely masks any wildlife signal that might be there. The researchers suspected this came from nearby agriculture, either pig farms themselves, or perhaps the use of pig manure as fertilizer in the surrounding landscape, which then became airborne.
Wow. That really highlights how human activities can interfere with this kind of sensitive detection. The noise can drown out the signal.
Absolutely. It's a critical constraint, especially when trying to use eDNA in landscapes heavily influenced by human activity. Intense signals, like from livestock, can potentially swamp the subtle signals from wild species.
And going back to the primers, the 16S for mammals and 12S for vertebrates, you said both were essential. Did they actually detect much overlap between the two?
Surprisingly little, actually. The 12S primer was responsible for capturing most of the bird and amphibian diversity. The 16S primer, while intended for mammals, was the one heavily impacted by pig DNA, but it did pick up most of the mammal diversity besides the pig signal. If they had relied on just one of those primer sets, they would have missed a huge chunk of the biodiversity they recorded.
So the lesson there is, don't put all your eggs in one genetic basket. Use multiple complementary searches.
Definitely. For robust airborne eDNA surveys, using multiple primer sets seems absolutely mandatory to get a comprehensive picture across different taxonomic groups.
They also did some modeling to figure out where the air itself was coming from, right? Using something called HYSPLIT.
Yes, HYSPLAT modeling. It's a tool used to calculate air parcel trajectories, basically. It backtracks where the air that arrived at their samplers had been previously. And the results were interesting. While most detected species were clearly local, the model showed that some of the air hitting their samplers had traveled considerable distances, potentially from elsewhere in Denmark or even other parts of Europe.
So theoretically, you could detect DNA from something hundreds or thousands of kilometers away.
It's possible, yeah. It's a reminder that you're sampling bioaerosols carried on air currents, and those currents operate on regional and even continental scales. It raises fascinating possibilities for tracking things like migratory pathways or maybe even the long-range dispersal of pathogens or allergens, but it also means you need to be cautious interpreting purely local signals.
Okay, so looking ahead, this was a groundbreaking proof of concept. What improvements do the researchers suggest to make this a more standard, reliable field technique?
Well, several things. One is finding ways to increase the amount of starting DNA, maybe simply by sampling for longer periods than just 3 days. More sampling time, potentially more DNA capture. They also suggest optimizing lab workflows, perhaps reducing the number of PCR steps if possible, to minimize errors and maintain the integrity of the low-concentration DNA.
And dealing with that randomness, the stochasticity.
Crucially, they emphasize the need for more replication. That means more samples collected in the field replicates, and running more parallel PCR tests on each sample in the lab, technical replicates. More replicates help you statistically filter out the noise, identify consistent signals, and get a much more robust and reliable picture of the actual biodiversity.
Makes sense. Build confidence through repetition.
Exactly. But the bottom line is, this study successfully demonstrated that airborne eDNA works for terrestrial vertebrates in nature, putting them alongside plants and insects where similar successes have been shown. It really positions airborne eDNA to become a key part of the toolkit for comprehensive biodiversity monitoring on land.
It really is a remarkable thought, isn't it? That the air itself, the stuff we breathe, holds this detailed, constantly updated inventory of the living world around us.
It absolutely is. And it pushes us towards some, well, perhaps slightly unsettling future thoughts, too. We saw how weather, things like rain and wind, dramatically affects where this biological material ends up. So think about the implications of that atmospheric transport. If animal DNA can travel like this, what about other things? How far could markers for pollution travel? Or, maybe more concerningly, markers for emerging infectious diseases carried in these bioaerosols?
You mean, could this technology become a tool for global surveillance of environmental health or even public health threats?
It's certainly a possibility down the line. Before science fully grasps how to track origins and maybe mitigate spread, this technology reveals just how interconnected things are via the atmosphere. It's not just about counting badgers in a forest anymore. It potentially opens a window onto much larger scale environmental processes. Something to think about.